All right, welcome everybody to lecture number 2, which is a discussion of both pH and buffering systems. So in this lecture, we'll start off with a very quick review of the units of concentration used in chemistry, so molarity. This should be a review from previous chemistry courses you've taken, but I just want to go over it because it's going to be necessary to answer some of the questions on the exam. Then we'll move into what pH is and how to determine the pH of a solution. So this will introduce us to equation number 1 of 3 equations that you're going to need to answer basically any pH or buffer question that I ask you on the exam. So it'll be how to determine the pH from a proton concentration or the proton concentration from the pH. Next, we'll move on to equation number 2 that you need to know, which is the ionization of water. And this will allow you to determine the proton concentration if you know the hydroxide ion concentration in solution and vice versa. And lastly, we'll talk about the ionization of acids in a solution, and we'll introduce you to equation number 3, which is the Henderson-Hasselbalch equation. And lastly, we'll round out the lecture with a discussion on buffers and buffering systems. So these are ways that you can regulate or moderate the pH of a solution. And so this is important both for biochemical experiments as well as your general physiology. Your body needs to be able to have a buffering system that will control the pH of your blood and keep it at a set level. So with that, let's launch into lecture number 2, pH and buffers.
All right, so one thing I want to go over very quickly is to review the units of concentration, which is molarity. So you probably covered this in other chemistry courses that you've taken, but it's going to be necessary to answer some of the questions on the exams and the assignments, and it's also just going to be very helpful to know as we move through our study of biochemistry. So the unit of molar concentration is 1 molar. So molar is the unit of actual concentration. And what that means is 1 molar would be 1 mole in 1 liter. So 1 mole is 6.02 times 10 to the 23 atoms of a pure molecule. So the mass of 1 mole of a molecule is going to be the molecular weight of a specific molecule or substance in grams. So this is going to be different for each different molecule. So for instance, the molecular weight of water is 18 grams. So oxygen is 16, and then hydrogens are roughly 1. So you have H2O, so that equals 18. So that's the molecular weight of water. So 1 mole of water is going to be 18 grams. And then 1 liter of water, we know, weighs 1 kilogram or 1,000 grams. So 1 liter of water, if you take 1,000 divided by 18, so 1 liter of molecule contains 55.55 moles of water. So the concentration of water is 55.55 molar. It's a very big concentration. Now, most biological molecules exist in much smaller concentrations than 55.55 molar. So you really wouldn't be working with a 55.55 molar solution when you're looking at a specific biomolecule. So it's going to be important for you to be able to convert between different units in this course. So just quickly go through that. So 10 to the 0 molar equals 1 molar, of course. 10 to the negative 1 molar would be 0.1 molar, which can then be converted into 100 millimolar. Just moving down the line, 10 to the negative 2 molar would be 0.01 molar, which would equal 10 millimolar, and so on. So you can see that when you're dealing with 10 to the negative 2 molar, it's a lot easier to just use the millimolar designation for that solution. But we're going to be using these interchangeably, so it's important that you study these a bit and figure out how to convert between the two. And so if even moving further down, 10 to the negative 6 molar would be 0.000001 molar, so that's not a very effective form to use that. You could use 0.001 millimolar, but even that's a lot of sort of painful. So we might just revert to 1 micromolar. So you need to be able to convert between micromolars and millimolar. And then moving even further, so if you're dealing with something like 10 to the negative 9 molar, you can convert that by saying 1, 2, 3, 4, 5, 6, 7, 8, 9. And that would equal 0.000001 millimolar, which even when you're getting to micromolar is a bit cumbersome. So 0.001 micromolar, you should actually probably use something like 1 nanomolar. So please take a little bit of time, make sure that you're comfortable converting through these things. So I'd maybe just pick one of these and say how many what is it in millimolar, micromolar, and nanomolar, and just make sure that you're comfortable converting between those. So now that we've covered the units of concentration, we can start talking about pH. And so pH is a very important parameter in biochemistry, and that's all basically most of what we're going to talk about today is going to be involving pH. So pH, put simply, is a measure of proton concentration. So protons are signified by H+, so they're just a hydrogen atom, and they have a positive charge. So you can see here that water, H2O, can break down in a reversible reaction into protons and H+ and OH negative, which H+ I'll refer to as just protons or a hydrogen ion, I might say that, or just H+, and then OH negative I would call a hydroxide ion or just OH negative. So pH is a measure of proton concentration. So this brings us to our important equation number 1 that you're going to need to know to do well in the assignments and the exams, which is pH equals the negative log base 10 of the proton concentration. So pH is a negative logarithmic function of the amount of hydrogen ions in a solution, or protons in a solution. So you could look over here. So pH goes on a scale from basically 0 to 13 or 14. And you can see it's directly related to the proton concentration in molar form. So that's going to be where you're going to have to convert some things. So a pH of 0 would mean that there's 1 molar of protons in that solution. pH of 1 is 10 to the negative 1. pH of 2 is 10 to the negative 2, and so on and so forth. And please, in your mind, just remember that 10 to the negative 10 is a much smaller number than 10 to the negative 2. So a pH of 2 is going to have a lot higher proton concentration than a solution with a pH 10. And of course, I mentioned pH is an important factor of biological solutions, and controlling pH is crucial for biological systems. So buffers are sort of chemical compounds that are able to control or regulate the pH of a solution. And you'll see how from this chart here, some pHs of biological solutions. So just starting in the middle, so human blood plasma is around pH 7. So when we talk about physiological pH, oftentimes we're just talking about blood plasma. We're just talking about pH 7 because that's mostly what your body is sort of made up of. You can see that there's other biological compounds that are not at pH 7. Including the human gastric contents, so what's in your stomach, that's going to be around between pH 1 and 2. So there's going to be a very high concentration of protons in your stomach. Cow's milk is basically neutral, but slightly more acidic. Human pancreatic juice is somewhat neutral, but a bit more basic. So it's important to understand that your body is not, different parts of your body can have different pHs depending on their function. But when we talk about physiological pH as sort of a shorthand, we're talking about human blood plasma, and we're talking about it being at around pH 7. So now we can actually already move into some of the example questions that you're going to need to know to do the exam. So one of the questions I can ask you is, what is the hydrogen ion concentration or proton concentration of a solution with pH 6. So we already know how to answer that problem. So the equation that we would need to use is our equation number 1, which would be pH equals the negative log at the base 10 of the proton concentration. Now, we already know that the pH is 6, so we can enter that into our equation. So 6 equals the negative log base 10 of the proton concentration. Now, this trips up some people on how to convert this, basically, to figure out the hydrogen ion concentration. But first, it's pretty simple, actually. So first, what I would do is get rid of this negative sign on this side of the equation. So we can say negative 6 equals the log base 10 of the proton concentration. And then I just do a quick arrows. You can look up online and if there's a different way you want to do this. But then you could just take the 10 in front, the 6 behind. So this would be 10 to the negative 6 equals the hydrogen ion concentration. So then we can rewrite this in a better form. So the hydrogen ion concentration would equal, let's say, 1 times 10 to the negative 6 molar, because this is going to be in molar. So this would be our answer to this question. So very simple, but it's good to just go through these. Another question I could ask you is, what is the pH of a solution with a hydrogen ion concentration of 1 times 10 to the negative 11 molar? So now I'm giving you the hydrogen ion concentration and asking you for the pH. So again, we only need that equation, pH equals the negative log base 10 of the proton concentration. And these brackets, of course, I didn't mention it earlier, but these brackets mean concentration. So we can put in our value, pH equals the negative log base 10 of 10 to the negative 11 molar. And then this one is actually super easy because you can just plug it into your calculator and you get the answer is pH equals 11. So that's the answer. Very, very easy to do. Okay, so question number 3. This one's pretty much the same, except I'm going to ask you to convert from millimolar to molar. So the question is asking, what is the pH of a solution with a hydrogen ion concentration of 0.01 millimolar? So again, we're just going to use that same equation we were using before. I really want to drill this into you. So pH equals negative log 10 of the proton concentration. But this proton, so we want to plug in our number into the proton concentration, but remember the proton concentration relating to pH has to be in molar, and right now we have it in millimolar. So there's a few different ways you can do this. I do it this way, where I say, oh, I want to go from millimolar to molar, so 1, 2, 3. So you can actually multiply it by 10 to the negative 3. And so that ends up being 1, 2, 3, 4, 5. So 0.01 millimolar equals 1 times 10 to the negative 5 molar. So now we can plug that into our equation. pH equals the negative log base 10 of 10 to the negative 5 molar. And then you can just plug that into your calculator. And you know that the pH of this solution with the proton concentration of 0.01 millimolar is at a pH of 5. So bear with me. These may seem pretty easy for you, but if you can master these, you're going to do really well on the exam. All right, so we kind of mentioned earlier, but now we're going to discuss in more detail the ionization of water. So just water in a solution will ionize to a small extent in that the H2O of water will convert in a reversible reaction to proton concentration or hydrogen ion concentrations, H plus, and OH negative, hydroxide ions. So the equilibrium constant for the dissociation of water, so this ionization of water, will be called KEQ. And so KEQ equals the proton concentration multiplied by the hydroxide ion concentration over the concentration of water. And this equation can be simplified to give the ion product of water. So the ion product of water is designated as Kw, and so that equals KEQ times the water concentration, and this is just done by rearranging this equation up here, and that will equal the proton concentration times the hydroxide ion concentration. So we actually know what this is. So at normal conditions, so 25 degrees Celsius, which is room temperature, and 1 atmosphere, this gives us an important equation number 2, which is the ionization of water equation, which is that the proton concentration and the hydroxide ion concentration equals 1 times 10 to the negative 14 molar squared. So I believe they derived this in your book, but you just need to know this equation, and that is the ionization of water equation. And what this allows us to do is to determine the proton concentration from if we know the hydroxide ion concentration and vice versa. So this will bring us to example question number 4. I'm going to do a lot of questions on these basically to save us all time later, because these are the type of things that you need to know by the end of the lecture to do well on the exam. So in this question, it's saying a solution contains a proton concentration of 1 times 10 to the negative 3. So what is the hydroxide ion concentration? So of course, we know the equation we need. So the proton concentration times the hydroxide ion concentration equals 1 times 10 to the negative 14 molar squared. Okay, so if we rearrange that, so we know the proton concentration, so we can write 1 times 10 to the negative 3 molar times the hydroxide ion concentration will equal 1 times 10 to the negative 14 molar squared. So if we rearrange this, because we want to know the hydroxide ion concentration, the hydroxide ion concentration will equal 1 times 10 to the negative 14 molar squared divided by the proton concentration, which is 1 times 10 to the negative 3 molar. So if you plug that into your calculator, or just do it in your head if you really want to impress yourself, you can get the hydroxide ion concentration, which would equal 1 times 10 to the negative 11 molar. So easy peasy, but it's good to go through these. So now we have another question that's asking us to push a little bit more. So a solution contains a hydroxide ion concentration of 1 times 10 to the negative 9 molar. So what is the pH of the solution? So for this, it's actually asking us 2 different questions, so we need to do 2 different equations. So to know the pH of the solution, we first need to know the proton concentration. And we can know the proton concentration because we know the hydroxide ion concentration. So the first equation that we're going to use is the ionization of water equation. H plus multiplied by OH negative equals 1 times 10 to the negative 14 molar squared. So to get the proton concentration, we will rearrange this equation. So the proton concentration equals 1 times 10 to the negative 14 molar squared divided by what we were given as the hydroxide ion concentration from the question, which is 1 times 10 to the negative 9 molar. So if we plug this into our calculator, we can see that the proton concentration is going to be 1 times 10 to the negative 5 molar. So now, but we need to know the pH of the solution, so we can use our equation number 1. So pH equals the negative log base 10 of the proton concentration, which we know now is 1 times 10 to the negative 5 molar. And if you put that into your calculator, you can get the answer, which is the pH equals 5. Okay, and we're going to do one more question. Like I said, I really want to drill these questions into your head, so let's just keep going with anything I could possibly ask you. So a solution has a pH of 10. What is the hydroxide ion solution? So we know that we can determine the hydroxide ion concentration from the proton concentration, and we know that we can derive the proton concentration from the pH. So not too difficult. So we'll start out with our first equation, pH equals the negative log base 10 of the proton concentration. So our pH is 10, of course. So 10 equals the negative log base 10 of the proton concentration. Let's get rid of that negative sign. So negative 10 equals the log base 10 of the proton concentration. And then we'll do this little trick, bring that in front, that behind. So and just go ahead and rewrite it. So the proton concentration equals 10 to the power of negative 10. And of course, that is in molar. So now we can use the ionization of water equation to figure out the proton concentration. And to save us a little bit of time, I'll just go ahead and write it after sort of rearranging the equation. So OH will equal 10 to the negative 14 molar squared over the proton concentration, which is 1 times 10 to the negative 10 molar. So your hydroxide ion concentration is going to be 1 times 10 to the negative 4 molar. Now, one other thing I do want you to check when you're doing these types of equations is that it makes internal logic, that it makes sense, your answers. So we know the solution has a pH of 10, which means that it's going to have more hydroxide ions than it's going to have protons. So because it's above pH 7, and that's sort of the delineation. So below pH 7, it's going to be acidic and have more hydrogen ions concentration. And above pH 7, it's going to be basic and have more hydroxide ion concentration. And of course, 1 times 10 to the negative 4 molar is a much greater number than our hydrogen ion concentration, which is 1 times 10 to the negative 10 molar. So it makes internal sense. So please check that before you click submit on your exams and assignments. All right, so now we're going to have a greater discussion of acids and bases, because you will encounter different acids and bases in when you're thinking about biochemical molecules. So acids ionize to form a proton and a conjugate base. So you have an acid here. There's going to be a reversible reaction where that acid will be deprotonated, basically release a proton, and then it will be left as a conjugate base. So an example of this would be something like acetic acid, which is a carboxylic acid. So it has this hydrogen group here. So acetic acid will ionize to form a proton and what's called acetate, which is its conjugate base. So the proton has a positive charge, conjugate base will have a negative charge. And it's really easy, so I can give you all sorts of different examples of these. So, but when the conjugate base of the acid is going to be an 8. You'd add 8 to the end of it. So if I say lactic acid, the conjugate base is going to be lactate. Formic acid, the conjugate base is going to be formate, and so on and so forth. So that's an easy shorthand you can use to remember this. Now the opposite, if a base accepts a proton, the result would be called a conjugate acid. So you have a base plus a proton. They're going to combine to form a conjugate acid. So it would just be basically the reverse of the previous equation. So acetate plus a proton can combine to form acetic acid. And it's important to understand that different acids have a different tendencies to ionize. So if you take acetate or acetic acid or lactic acid, they're going to lose those protons at sort of a different rate or a different value between these different acids. And that's just a characteristic of each different chemical as it exists in nature based on its chemical properties. So the ionization equilibrium of a weak acid, which is an acid that doesn't readily get deprotonated, can be given by this reversible reaction, which is HA. So that's sort of how we would refer to a weak acid as a shorthand. So this represents the hydrogen ion, and this represents the acid. And that will ionize to form, of course, a proton and the conjugate base, which is signified by A negative. So we can form an equilibrium constant for these types of reactions by using this equilibrium constant called Ka, or the equilibrium constant for acids, A for acids. So Ka will equal the ratio of the proton concentration and the conjugate base concentration, A minus, over the concentration of the acid. So this is our equilibrium constant for the ionization of different acids. So one thing we can determine from this is that the larger the Ka, the stronger the acid. So the more readily, if this is a high number and this is a low number, so the stronger the acid is actually going to dissociate. Or it's going to dissociate more readily in water. So this gives us a way to describe how readily an acid will ionize or become deprotonated in a solution. So strong acids will dissociate very readily in water and have a very high Ka. So for example, something like hydrochloric acid is going to readily dissociate in an aqueous solution in water to a proton and a chloride ion, so Cl negative. Whereas weak acids, which we'll encounter more in this course, are going to dissociate less readily in water. So acetic acid is going to dissociate less readily than acetate and thus have a lower Ka. There's going to be there's going to be more of an amount of this Ha and less of an amount of the proton and A minus, which in this case would be acetate. So really this Ka equilibrium constant gives us a way to describe this ionization ability of different acids, which is just a property of each different acid in nature. So, but we need an equation that will allow us to describe the relationship between the pH and the ratio of the acid and conjugate base in a system. And this is especially important for buffers. And so that ratio of acid to base describes how readily that weak acid is going to dissociate or ionize. So our ionization equation can be manipulated to give 1 over the proton concentration equals 1 over that dissociation constant, Ka, multiplied by the conjugate base concentration to the acid concentration. Now, if we take the logarithm of both sides of this equation, it gives us log to the 1 over proton concentration equals the log to 1 over Ka plus the log of the conjugate base over the acid concentration. Now, this is very helpful because the log of 1 over H plus is basically the same. It is the same as negative log to the proton concentration. And so that's the same as pH. And then this log of 1 over Ka now gives us a new value that we can play around with called pKa. And we're going to keep the last part of this equation because we want to have part of an equation that talks about the ratio of the acid base in a buffer system. So this rearrangement and taking the logarithm gives us our important equation number 3, which is the famous Henderson-Hasselbalch equation. So the Henderson-Hasselbalch equation says the pH equals the pKa plus the log of the conjugate base A minus over the acid HA. So this is going to be the third equation that you're going to need to know for this course and to do well on the exams. So as we said, now this Henderson-Hasselbalch equation, that's perfect for describing the relationship between the pH and the acid-base ratio. So here we have the Henderson-Hasselbalch equation written right here. pH equals pKa plus the log of the conjugate base concentration over the acid concentration. And this gives us 3 clear insights into how these weak acids work in a solution and ways for us to describe them. So 1, and this is very important for you to remember. So the pKa is the pH at which the acid concentration equals the conjugate base concentration. So the pKa is the pH at which the acid is half protonated or dissociated or ionized in a solution. And this makes perfect sense because if we look at our equation, so pH equals pKa plus the log of the conjugate base over the acid concentration. So if we make the conjugate base and the acid concentrations the same, this is going to be log of 1, which equals 0. So log of 1 equals 0. So that would mean pH equals the pKa. So this pKa is again just it's a really useful term for describing the ability for a weak acid to become deprotonated in a solution, and there's going to be a different pKa for each different type of weak acid. So we have acetate again right here, and acetate over here. So the pKa of acetic acid is going to be 4.8. And so because that is the point at which the acid and the conjugate base are going to be present in equal concentrations. Now, 2 more important points from this, which we can already see, is that when the pH is less than the pKa, this acid species is going to dominate. So it's going to be there's going to be more of this acid species in the solution below the pKa. And then third, when the pH is greater than the pKa, the conjugate base form, the deprotonated form, is going to predominate. And you can basically play around with these numbers yourself and just plug them in, the A minus over the HA. If the A minus, the log where the conjugate base is going to be in a higher concentration than the acid, you're going to add, it's going to be a positive number, and the pH is going to be higher than the pKa. If the acid is a greater number than A minus, you're going to get a number below 1 for this log, and it's going to be a negative number, and the pH is going to be below the pKa. So this is a very important relationship to understand and on what pKa is and how it relates to this Henderson-Hasselbalch equation. So this will bring us to another example question. I'm going to go through 3 more questions, and then we'll be done with questions, but it should be all the information you need to answer any question, like I said. So this question is asking us, so it's formic acid, which is CHOOH, is a weak acid found in Hymenoptera venom, so ant bee venom. And formic acid has a pKa of 3.75. So at pH 2, which form of formic acid will predominate? So will the acid be the dominant molecule? Will formate, which is the conjugate base, be the dominant molecule? Or will they be present in equal concentrations? So you don't even have to do any math for this. You can, if you want, you can just make up a ratio where one is in a higher concentration than the other and plug that into the Henderson-Hasselbalch equation. But we already know that the pH of 2 is below the pKa. So then our correct answer is going to be that the acid form, formic acid, is going to be the dominant molecule in this solution. So that type of question should be pretty easy if you just take some time and learn the concept of pKa and the Henderson-Hasselbalch equation. Okay, so in the question number 8, so in the previous example, there was a solution of formic acid, which has a pKa of 3.75. That's just a fixed property of formic acid. So, and the solution is at pH 2. What would actually be the precise ratio of formate to formic acid in the solution? Now, we know we can answer this by using the Henderson-Hasselbalch equation. So, pH equals pKa plus the log of this ratio A minus over HA. It's the conjugate base over the acid. So we already have a lot of information that we can plug in. We know the pH, so 2 equals the pKa, which is 3.75, plus the log base 10 of A minus concentration over HA concentration. So we will subtract 3.75 from each side. So this gives us 1.75 equals the log base 10 of A minus over HA. So now if we just do our conversion here, it's my little trick, but you guys can do it whatever makes most sense in your head. So 10 to the negative 1.75 equals the ratio of the conjugate base, formate, to the acid, formic acid. And if we plug this 10 to the power of 1.75 into our computer, or into our calculator, we can find that A minus over HA, so that ratio between the two equals 0.018. And again, this makes internal logic that you would have much less of the conjugate base compared to the conjugate acid, and then thus have a ratio below 1. So that's a very good example of a question that I could ask you on the exam. All right, one final question, but this allows us to determine a different type of part of the equation. So in this question, it says a solution consists of 0.8 molar sodium formate, so the conjugate base, and 0.2 molar of formic acid, the acid. And we know that it has a pKa of 3.75. So what is the pH of this solution? So we know that we need to use the Henderson-Hasselbalch equation. So pH equals pKa plus the log base 10 of conjugate base over the acid concentration. So we already have a bunch of the information. So we don't know the pH, but we know the pKa is 3.75. And that's plus the log base 10 of 0.8 over 0.2. And those are both in molar already, so you don't have to do any conversions because it's already in molar. So now we can simplify this a little bit more. pH equals 3.75 plus the log base 10, and 0.8 over 0.2 is 4. So pH equals 3.75 plus the log base 10 of 4, which turns out to be, if you plug that into your calculator, 0.6. So the pH will equal 4.35. And again, this makes internal logic because we know that we have more of the conjugate base than we have of the acid. And so we already know that the pH is going to be higher than the pKa, which it is. So the pH is going to be 4.35. So now we'll finish up with a short discussion on buffers. So buffers are basically solutions that will resist changes in pH. And these buffers are made from a weak and acid and conjugate base pair. And these will resist changes of pH in a solution. So that is the definition of a buffer. So if you have a weak acid and you add hydroxide ions, it's a reversible reaction where you're basically just going to have the conjugate base plus water. And so you're not really having a change in the free proton concentration in the solution. So the weak acid in this solution will counteract changes in the proton and hydroxide ion concentration. If you add more of them to the system, they're going to counteract changes to the actual pH because they're able to actually absorb protons that would otherwise be put into the system. So here's a little diagram here, again, using our friend acetic acid. So you can see that you have a solution that's at pH 2. And so, of course, that's lower than the pKa. So you're mostly going to have this acid. But if you also have the conjugate base in there, it's going to work like this, where if you add hydroxide ions on the solution, it's very quickly the pH is going to very quickly rise. But around the pKa, it's actually going to allow buffering of the solution. So you're going to have resist the change in pH as you're adding hydroxide ions. So you've already added way more hydroxide ions, but the pH has actually only changed to a very small extent. So this is how buffer systems work. And this would be called basically the buffering region, because above or below this region, the buffer system isn't able to resist the addition of protons or hydroxide ions. So it's going to have a very quick change in pH. So take some time, try and think about these types of systems. And yeah, if you have any questions on that, let me know. And we sort of saw it in the last example, but buffers are most effective near their pKa. So that's an important sort of concept to understand. They're going to be most effective when that ratio of acid to base is going to be closer to 1-to-1 ratio. So here we have a lot of different examples. We have acetic acid here. You can see very quickly goes up in pH, and then it's in its buffering region and goes up. But this works for other buffering systems too, including this system with phosphoric acid and sodium phosphate. I forget what it's called, and then ammonium as well. So all these different buffers. The important part from this is going to be basically that they're going to be most effective at or near their pKa. And the last topic we're going to talk about today is that buffers are essential to biological systems. So the pH of your blood, which we mentioned needs to be maintained around pH 7, physiological pH, this is buffered by the conjugate acid-base pair of carbonic acid and bicarbonate. So carbonic acid is shown here. It's H2CO3. And then bicarbonate is the conjugate base of carbonic acid, which is HCO3 with a negative charge. So the CO2 produced by normal aerobic respiration in our body will actually react with water to produce a weak acid, carbonic acid. So CO2 plus H2O is a reversible reaction, but it'll result in the production of carbonic acid. So then carbonic acid will then ionize to produce both a proton and bicarbonate. So this combination of a weak acid and its conjugate base, carbonic acid and bicarbonate, is what allows our body to keep our blood plasma at a pH 7. If it goes way higher than that or way lower than that, your cells cannot sustain life and you will die. So this is a very important part of biochemistry.
All right, so that concludes lecture number 2, which is pH and buffers. And there are a few take-home summary points that I really want you guys to understand and to study. So one is that pH is a measure of the proton concentration in a solution. And you'll need to know how to use the first equation, which is pH equals the negative log of the proton concentration. So if I give you proton concentration, you should be able to figure out what the pH is and vice versa. Second, you need to know that acids are proton donors and bases are proton acceptors, and understand the reversible reactions between the two. You need to understand that water ionizes to a small extent, and to understand how to use equation 2, which is the ionization of water equation. So that is that the proton concentration times the hydroxide ion concentration of a solution is equal to 10 times -4 to the power of -14 molar squared. So if I give you the hydroxide ion concentration of a solution, you need to be able to figure out the proton concentration and vice versa. Next, you need to understand equation number 3, which is the Henderson-Hasselbalch equation, which describes pH as a function of the conjugate base to acid ratio. And for that, you also need to understand the concept of pKa, and to know that pKa is the pH at which the conjugate base and the acid are present in equal concentrations. So if the pH is below the pKa, then the acid is going to be the predominant species in that solution. If the pH is above the pKa, then the conjugate base is going to be predominant in that solution. So please play around with that and understand that concept. And lastly, you're going to have to know how buffering systems work, and that biological systems use the carbonic acid bicarbonate system as a buffer. So I hope you enjoyed the lecture. Please let me know if you have any questions.